So continue our journey for the next question of 103, asking Neet UZ 2020. Yes. So we have already discussed, yes, question number 105 with its answer 2. We discussed 101 with the answer 3 with explanation. Now we are going to discuss question number 103. Yes, this question you all know that it is from your class 11, NCERT class 11, base question from plant physiology, and especially your very interesting chapter, respiration. So the question is, the number of substrate level phosphorylation, in one turn of citric acid cycle. Right now, so this citric acid cycle means to say it is talking about, yes, Krebs cycle. Fine. So you all know about the Krebs cycle, and question is asking about if Krebs cycle is being turned once, then you have to find out the number of a reaction called phosphorylation. And this is not the common phosphorylation, this is a special phosphorylation called substrate level phosphorylation. As we all are here to explore biology from A to Z, so we have to understand first two important terminology. First one is the phosphorylation. By name it is very much clear. Phosphorylation means to say addition of phosphorus, means to say what happened, as a result there will be formation of ATP. So this phosphorylation is of two types. You all know that it may be substrate level phosphorylation, what the question is asking in 103 question, substrate level phosphorylation or substrate phosphorylation. Another one is known as chain level phosphorylation, right? So the chain level phosphorylation, what happened here? Here either NADH2 or FADH2, right? Now FADH2, right, will oxidize, and through ETS, electron transport system, they will release you ATP. So you are not going to get ATP directly, rather the ATP will be generated through ETS only. That's why it's called chain level phosphorylation. But when you are getting, suppose A is there and that giving the product B, and you are getting that very A, right? This A is giving to your B, and during this process if you are going to generate ATP directly without ETS, then it's called what? This is called substrate level phosphorylation. For an example, remember in very simple, in glycolysis, remember what happened last step? Remember, we have phosphoenol pyruvate, and that phosphoenol pyruvate gives the pyruvic acid. So this phosphorus is releasing, so that phosphorus is taken by ADP, and that ADP with this phosphorus, what happened? It will generate ATP. So this is the direct or substrate level phosphorylation. Substrate is providing phosphorus and there is a formation of ATP. That's why it's called substrate level phosphorylation. But I'm not talking about, this is a common concept about the phosphorylation, what the question was concerned. But we have to discuss this substrate level phosphorylation not in glycolysis, rather in the Krebs cycle. My dear friends, remember the Krebs cycle. What happened? During the Krebs cycle, what happened? There is a formation of, as I told you, NADH2, and FADH2, right? So once FADH2 formation is there and thrice NADH2 formation is there, as well as, right, so these are the chain level phosphorylation. But when you will talk about the GDP, that GDP is going to form GTP, and through the coupling reaction there will be formation of ATP. So what you are finding again, you are finding that how that GDP or this GTP or through the coupling reaction ATP is going to be formed directly from the substrate, and this is called substrate level phosphorylation. So we have a single step where it occurs. So what is the answer? Answer is 2, 3, 0, 1. So exactly it's 1. Okay, so as I told you, this is again direct question, very easy question, and directly associated to your NCERT line. How? Take the glimpse. Yes, this is the very simple explanation of NCERT. This is the citric acid cycle, right? In this citric acid cycle, that the Krebs cycle, what happened? See, this cycle is occurring once, right? And see, this is the acetyl coenzyme. Okay, and this acetyl coenzyme is going to form citric acid with oxaloacetic acid, and finally alpha ketoglutarate acid is there, and we form here succinic acid. During this process there is a formation of this is the ZTP, and it will only form through the substrate level phosphorylation. So what we are finding, see how we have, this is the FADH2, this is the chain level phosphorylation. See, this is again NADH2 substrate, this is the chain level phosphorylation. NADH2 chain level phosphorylation, NADH2 chain level phosphorylation. So as I told you earlier, thrice NADH2, 1, 2 and 3, and once FADH2, right? This is also through the chain level phosphorylation, and again once this is the substrate level phosphorylation, GTP will forms right now. But I will also like to explore you to understand this all the Krebs cycle, that there we have this pyruvic acid, we have glucose, that glucose will form pyruvic acid, this is the glycolysis, it will form acetyl coenzyme A, this is the oxidative decarboxylation will be there, and then there will be citric acid, right? So just remember a mnemonic, what? Cricket Club of India, OK Sachin, remember, Sa, Fa, Ma, Oe, Oe. So this is a very good mnemonics to remember the Krebs cycle. As I told you, citric acid, right, citric acid, isocitric acid, oxalosuccinic acid. Yes, this is the alpha ketoglutarate acid, this is the succinyl coenzyme A, this is the succinic acid, fumaric acid, malic acid, and oxaloacetic acid. Right now, so this is the cyclic process we are going to form the whole elaborated compound we have, right? So see, this is the 2 carbon compound, fine, and this is the 4 carbon compound. So 4 plus 2, yes, we have first, this is the 6 carbon compound, and this 6 carbon compound is the citric acid. That's why it is called citric acid cycle, right? And since it was discovered by Hans Krebs, so it's also known as Krebs cycle. So see, this is again 6 carbon, this is again 6 carbon, this is again 6 carbon, and once you will find here what? Decarboxylation will be there. Again you see this is there, again decarboxylation will be there. So you find these are the 6 carbon compound, one we have the 5 carbon compound, and here these all are of the 4 carbon compounds, right now. So what happened? You will find during this process here you will find this the exact position we have right now. This is the exact position where you are going to generate what? this is your GTP. So I think you remember here that how we are going to form. This is the citric acid and oxaloacetic acid, right? So here what happened, we have also a compound, and there is also some enzymes are involved. To remember the enzymes, you must remember this trick. In class I used to say, remember SAD, TD, I think all of my students are very known of my this mnemonics. Okay, this is the SAD, S-A-A-D, remember S-A-A-D-D-D. Thrice we have SAD, then T-D-F-D, remember. Okay, so this is the synthetate, this is the aconitase, this is dehydrogenase, means to say there will be formation of NADH2, very easy to remember. Similarly, this is decarboxylase, that's why you are finding CO2 is releasing here. Again, this is DDC, that is dehydrogenase decarboxylase complex. So what happened? Decarboxylase, so CO2 is releasing, and here dehydrogenase, so there is also formation of NADH2. This is the thiokinase, and this thiokinase is going to form, what is the question? This is going to form ZDP to GTP, and through this coupling reaction it will form ATP. Again, this is the dehydrogenase and it will form FADH2. So once FADH2 will form. Again, this is the fumarate, and this is the dehydrogenase between malic acid and oxaloacetic acid, and this dehydrogenase will again form NADH2. So thrice you are finding NADH2, once you are finding FADH2, once you are finding GTP. So GTP is the substrate level phosphorylation, and this was the question. So I think for further also you are well versatile with the Krebs cycle. How you will remember the Krebs cycle? Yes, you can remember by this very good mnemonics. Yes, what is that? That is the Cricket Club of India. O K Sachin, Sa Fa Ma Oe Oe, right? And what is the enzyme? Enzyme is SAADDD, SAD, TDFD. Okay, so the D is either dehydrogenase or decarboxylase, right? So twice we are going to execute what? Decarboxylase, and you are finding 4 times, right? Now thrice again we are going to form this formation of NADH2, once FADH2. So I think you are very you are well known by this Krebs cycle, and further if you are getting any question associated to Krebs cycle, I think you will solve it very well. So finally I conclude the answer of question number 103 is what? 4 ones. So in citric acid cycle, Krebs cycle, the number of substrate level phosphorylation in one turn is 1. Okay, so thank you.